So hello everybody, and thank you for your participation to this really first webinar of the Marseille Institute. So as you know, we had to cancel our kickoff meeting, which was supposed to bring us all together for a scientific day of exchanges and discussion. And in the current context, it is essential for us to imagine new ways of meeting with the objective of knowing each other better. Thus, we decided to propose this new meeting, the Neurobinar, which will take place once a month on Thursday. And maybe we can have the next slide, Ingrid. Thank you. The next, so once a month on Thursday, and we'll allow 3 scientists from the Marseille Institute to present their work. So once a month, the program of the Neurobinar will be as follows. We will have first a presentation from 2 students from the PhD program who have accepted the challenge to present not their own work, but the work of a fellow from the PhD program. And then during a second part, a scientist will have a 20-minute slot to present his or her work. And today we will be listening to Rochelle Ackerley from LNSC, who will present her work on the generation of artificial touch. Of course, these 2 sessions will be followed by questions, and we suggest that you type directly your question or ask for the microphone via the Q&A at the bottom of your screen, and Gabrielle and Ingrid will connect you by turns. So let's start with our first session. And today, this is my pleasure to introduce the 2 brave PhD students who have accepted the game of the cross presentations. So these 2 students are Lucie Pepino from IBGM and Mikhael Azilinon from CRMBM and INS. So Mikhael will first present Lucie's work on pain modulation, and then Lucie will present Mikhael's works on cortical excitability and epilepsy. At the end of these 2 presentations, we will have questions for both of them. So I would like to thank again the speaker to have accepted to participate to the first Neurobinar. I hope you will really enjoy the program and that this Neurobinar will be the first of a long series of scientific exchanges within our community. So Lucie, Mikhael, the stage is yours now.
Thank you, Pascal, for the introduction. So I will just share my screen and start the presentation on Lucie's work. So we all have someone in our surroundings to remind us that girls are more resistant to pain than boys, which tends to provoke endless debates. Presenting Lucie's PhD project today is like setting fire to the powder keg because her PhD is entitled Sexual Difference in Pain Modulation, a possible link between sex hormones and pain modulation. During decades, most of the pain research has been carried on male rodents. Yet women and men experience pain differently. This suggests sexual dimorphic mechanisms involved in pain processing, modulation, and perception. Understanding the molecular and cellular mechanisms underlying sex differences in pain neurobiology is a very active topic in the field. So through her work, she is aiming to answer at least 2 questions. Is spinal modulation of pain sexually dimorphic, and do males and females activate the same spinal cord network in response to a same painful stimulus? In order to answer those questions, she used genetically stable mouse model. She then tried to evaluate the hormonal impact on pain, identifying in which part of the estrous cycle the mouse is during the experiment. And finally, she triggered the inhibitory pain system. To trigger the inhibitory pain system, she stimulates pain using a chemical pain model using formalin. So she measured behavioral pain. Oh, sorry. She measured behavioral pain answer by timing, and anytime the mouse expressed spontaneous pain behavior like licking, beating, flinching, and lifting the hind paw, as you can see on this video. Here she showed the classical male response to formalin injection, which is biphasic, with the first phase here with important pain behavior from 0 to 10 minutes, then an interphase where pain behavior decreases from 10 to 25 minutes, and finally, pain resumes in phase 2. In contrast to males, females seems to be segregated into two subgroups. Early phase 2, where pain resumes earlier than males during interphase, and late phase 2, where pain resumes later than males. The difference between females is apparently due to the estrus cycle, as formalin nociceptive response fluctuates across the estrus cycle, as you can see on this graph, where in green you have early and in red the late one. The inhibition of pain seems to be decreased in proestrus estrus females. Is that right, Lucie?
Yes.
So she explored the behavioral response of males and females to formalin. Her experiments prove that circulating sexual hormones are able to influence the inhibition of pain. She is now seeking to understand the mechanistic aspect to decipher the genes expressed differently according to the gender.
You cannot really say gender for mouse, huh? It's sex.
Why?
Because gender is the way you define yourself, and there's no way we can assess that in mouse. Huh?
Well, maybe there will be other questions on this. So now I will let you talk about my PhD.
Hi everybody, so I'm going to share my screen. Good, working? Okay. So before getting into Mikhael's PhD project, I wanted to ask you if any of you, I mean the virtual audience and the real audience, if you ever heard about sodium MRI and 3-day spectroscopy imaging techniques.
Yes, I did.
Yeah. Well, for me, I never heard of it before talking with Mikhael, so I'm going to try to present you why is he using that technique. I'm not going to get into the techniques because I'm no physicist, so please keep your technical question for Mikhael. So his PhD work is on sodium homeostasis and cortical excitability in human partial epilepsy. So in pharmacoresistant partial epilepsy, the only cure available is surgery. So they need to remove the part of the brain for which is responsible for epilepsy. But in order to remove the part of the brain, they first need to investigate where the epilepsy is coming from. So for that, they implant electrodes into the brain of the patient by using SEEG, and they record the activity, and it allows them to specifically localize the epileptogenic zone, the propagation zone, and the non-involved regions. But of course, a non-invasive and innovative method should be privileged. That's where Mikhael's work is stepping in, right?
Yes.
So for about 10 years now, Victor Gerza's lab and his team has been working on a computational framework the virtual brain that stimulates cerebral activity within and between regions using a neural mass model. Don't ask me to go deeper because I cannot go deeper in my explanation. And so they developed a specific model, the epileptor, that is able to simulate seizures thanks to the information on the structural connectivity and the clinical hypothesis from the doctors from the SEEG data. So the aim of Mikhael's PhD project will be for the first time to implement MRI data into this model. So for that, he's going to use a multimodal imaging approach using both, as I told you, sodium MRI and also 3D spectroscopy imaging. It will help him to localize the epileptogenic zones, and it will give him the homeostatic and metabolic abnormalities in cerebral tissue, and it will give him information, direct or indirect information on the cortical excitabilities. So briefly, the sodium MRI is going to give you the total concentration of the sodium within the brain. Mm-hmm. And the 3D spectroscopy imaging is going to quantify some metabolites, the N-acetylaspartate, choline, and creatine. Mm-hmm. So for his project, it's going to be the first time that sodium MRI is going to be used on epileptic patients. The first time that 3D spectroscopy imaging is going to be used on drug-resistant patients, and the first time that the two techniques are going to be combined together. So a very exciting project, huh? So I'm going to explain to you only one result because that's the only one I can more or less understand. So here you have the different zone that I told you about, the epileptogenic zone, the propagation zone, the non-involved regions. On the left side, you will have the sodium concentration. On the right side, you will have the N-acetylaspartate quantification. And every time in gray, it's the control, and the colored and black plots, it's the epileptic patient. So what it shows you is that apparently the sodium concentration seems to be higher in epileptic patients than in control, right? He's making a face. Not always. Most of the time. While for the N-acetylaspartate, it seems to be decreased in the 3 of the zones.
Okay, let's say that.
Let's say that, okay.
Not exactly this, but...
Mm-hmm. But what he told me was important to remember is that some measures are more sensitive to alteration within the epileptogenic zones, while other seems to be more sensitive outside of the epileptogenic zones. And that in that case, with focal epilepsy, damage is often diffused within healthy zones. Yes? Okay.
That's the best sentence you made since the beginning.
Mm-hmm. And so now that he's done with the multimodal imaging, he's gonna try to implement that into the virtual brain project, trying to find the right mix of measure to best inform the virtual brain. Mm-hmm. Okay. Yes.
So thanks a lot, Julie and Mikhael, for this presentation. Lucie, oh my God. I'm really sorry. So Lucie, thank you. Thanks a lot. So do not hesitate if you have any questions. There is one from Benoit Drouillas. So maybe Lucie, you can ask, I don't know if you receive it.
No.
So the question is, I don't know if I missed the information, but what's the age of the black cyst used by Lucie? Does that mean that the differences observed between male and female are not present in early stage of development? When can we consider the pain spinal network mature?
Okay, so the mice, sorry, I always make the mistake, that I'm using are between 8 and 12 weeks old. So they are mature mouse with a hormone level as adult. I mean, they're able to procreate, so they are adult mice. And then if it's not present in early stage of development, but I cannot really answer that because we did not test it. But as our main hypothesis is the fact that we have a hormone influence, because we did some hormonal dosage that we didn't show here. And it seems that estrogen and prolactin is influencing on the pain behavior of females. We can imagine that it will not be present in early stage of development because the hormone level in the females are rising during teenager, which is, I think, between 3 and 4 weeks old. And when can we consider the pain spinal network mature? Well, if you want to consider the hormonal level, we choose to use the adult mice, but the spinal network is mature. I think in my team, when they do electrophysiology, they use mice between 4 and 5 weeks old. So we can consider it's mature then. Mm-hmm.
So thanks a lot, Lucie. So we have another question for Mikhael. Can you remind the advantage of Na plus MRI? Do you have an idea about the mechanism underlying the sodium increase?
Thank you for this question. So first, the advantage of sodium MRI is to quantify the sodium in the brain in vivo, which is just not possible otherwise. And there is a lot of potential mechanisms that can underlie the sodium increase, and well, we are not really sure right now. But in the context of epilepsy, there is at least two main. Well, the increase should be seen because of an intracellular increase. That is our hypothesis. There is other possibilities, but it seems to be the major one. And this intracellular increase should be can be due at least to two main reasons, which are bad inactivation of voltage-gated sodium channels and an effect of sodium-potassium pump. There is these two reasons to an intracellular increase, which will be seen in the total sodium concentration increase. I don't know if it is clear, but that's my answer. And the last question was, how long does take a sodium MRI session acquisition? Well, on the 7 Tesla machine, so data that I processed, we made 3 runs of 10 minutes, so it will take 30 minutes, around 30 minutes, maybe 40 for all the calibration stuff and that.
Yeah, last one for me.
Okay. Does a higher rate of sodium in the brain means you are most likely to develop epilepsy? Is that why we use the sodium MRI to quantify that? Well, no. Actually, I'm just focused on drug-resistant epilepsy. So it's epilepsy that already well established. So we now want to use sodium MRI to investigate if the fluctuation of sodium can be a biomarker of cortical excitability or of the epileptic network. Actually, it's a technique that were never really used for epilepsy. There is only one paper on sodium MRI and epilepsy, which was made in our lab. So it's like something not a big hypothesis about how things should go in epilepsy with sodium concentration.
Okay, so there is another question for Lucie. Why do you use formaldehyde instead of other pain inflicting methods?
Okay, so we choose to use that specific model because we wanted to study the acute pain at first. And specifically, we wanted to trigger pain inhibition system at the spinal cord level. And it has been shown in the literature that in the formalin pain model, the interface is thought to be due to active inhibition by GABAergic interneurons at the spinal cord level. So that's why we choose to use that specific model to look specifically at the GABAergic inhibition system. Pardon.
So I'm sorry, we're gonna stop with the question, but I see there is 2 more for Mikhael, so he's gonna answer those questions in the Q&A space. And now we are gonna start with Rochelle presentation, and Boris Burl is going to present Rochelle.
Okay. Everybody, it's my great pleasure today to introduce the first speaker of the Neurobinar, of the first Neurobinar. The speaker will be Rochelle Ackerley. She defended a PhD in 2006 in Bristol. And then she was cognitive neuroscientist at Unilever for 1 year, from 2006 to 2007. She made then 2 postdocs, one from 2010 to from 2007 to 2010 at the somatosensory lab at University of Manchester. And then a second one from 2010 to 2017, Department of Physiology of University of Gothenburg. And since 2007, she got a CNRS position at the NNSC. And in 2019, she was awarded by a Consolidator ERC grant. And in 2020, she received the CNRS Bronze Medal. And now I leave the stage to Rochelle.
Thank you very much. I'll try and share my screen. Can everyone hear me okay? I'm guessing yes. And I'd just like to say a big thank you to the students as well presenting here today. They did really great presentations. Okay, so I'm guessing that everyone can see everything. So today I'd like to tell you about some of my research and also a little bit about the lab here in the LNSC. So we work in the Laboratoire de Neurosciences Sensorielles Cognitives, and that's situated in Saint-Charles. And we're actually made up of 4 teams, and we are directed by Beatrice Alessio-Lautier. We do a lot of different work here, and it ranges from all different types of work on sensory mechanisms all the way through to different cognitive processes as well. And we do this at various levels, and this is from cell biology to recordings and electrophysiology, both in humans and in animals. And we also do various different behavioral experiments, and this goes all the way up to tests in patients. So the first team I want to present is the Équipe Dynamique Neuronale et Audition, and that is led by Ono Norina, and they look at many different types of neurodynamics, but they use tinnitus as a model, and they look at the neuronal representations of both physical and perceptual sounds. And they also look at the plasticity associated with this, the modification of this sensory input. And then we also have the second team, which is the Équipe Physiopathologie et Thérapie des désordres vestibulaires. And that is led by Christian Chabert. And they mainly look at the generation, the encoding, and the transmission of vestibular information. And this includes the adaptive mechanisms that's involved in the restoration of especially auditory and vestibular syndromes. And they also are looking to optimize the methods to improve the diagnosis of vestibular disorders. And then next we have the Équipe Réhabilitation Sensorielle et Cognitive, and that is led by Beatrice, who leads the whole lab, and also by Liliane Borel. And they look at various different things to do with sensory interactions and your balance, and also cognitive interactions underpinning your cerebral mechanisms. And they also look at various different parts of rehabilitation. So this is all sorts of rehabilitation to do with your balance control, but also cognitive rehabilitation as well. And I'd just like to say that at the moment, maybe it should have finished by now, but we have Claire in the middle, who has just been defending her thesis. So I hope everything's gone very well for her. And then we come to the team that I am in, and it is the Équipe Corps et Multisensorialité. And this is led by Anne Cavenu-Diaz. And we are actually quite a big team. There's 8 statutaires, so permanent people, as well as 3 postdocs and 5 PhD students. And my group is part of this team here. But in general, we look at the multisensory contribution to body and self-perception, and also the neuronal encoding both in the periphery, which is more what I do, all the way up to neural coding in the cortex. And then we also look at adaptive plasticity in the mainly somatosensory system as well there. So I'd like to go on to talk about more of my work here. And the majority of my work looks at touch. And so I believe touch is very, very important to everyday life. And I like to say that it's a language that we understand instinctively. You don't have to learn how to understand to touch someone, to touch something. You don't have to learn how to do it. You just know. And so when we think about touch, it's normally using our hands to explore different things. And that's the duration of the touch, whether it's rough or how long it lasts, and if it's edges, if it's rough, things like this. But that's not just what touch is about, because there's all sorts of different dimensions here, such as softness and wetness and stickiness and texture. And then you've got temperature in touch, which is quite important as well. And then you can go all the way up to more of an emotional level in touch. So is it pleasant? Is it calming? Is it itchy, the touch, or soothing, or unpleasant even? And the majority of my work is on the fundamental level. And so you need to know about how the system works to then understand how you can treat different disorders and things like this, which I'll go into a bit as well. So how do you feel? How do you go about feeling things? Well, normally when you think about this, you think of exploration. So you'll go and explore your world, and then you can take it a step further to think about things like feeling different textures and the complexity that you feel in many different things all the time in your life. And this is what I tend to think of as discriminative touch. And so this is, you know something has happened, you know exactly where it is and what it is. However, you've got another side to touch, which is a lot less studied, but it's very interesting. It's an area that's really upcoming, and this is affective touch. And so affective can mean unpleasant, such as pain, but it can also mean pleasant touch, which I'll tell you a bit more about as well. And then you've got questions on the restoration of touch, which is a much more recent thing. And I'm going to tell you hopefully about the near future and potentially the slightly further away future and some ideas we have where we'd like to go. So when you think about touch, you think about the skin, of course, and you've got mechanoreceptors in your skin, and this is all over your body. So it's you can distinguish the skin between the glabrous skin, for example, of your hands where there's no hairs, and then the rest of the skin of the body where there's lots of different hairs. And you've got different types of mechanoreceptors that signal different things across the body. However, thinking about touch more generally, the skin is actually our largest sensory organ. In fact, it's around 2 meters squared, which is really very big. And I just want to put it in perspective a bit that we don't really know the exact number of receptors we have or afferents that send the information to the brain we have in the skin. So we think that we have maybe between 45,000 to into the millions of afferents that signal somatic sensation from the skin. But we don't know. But what we do know is there are about a 5% decrease in the number of afferents per decade of life. So, for example, in the hand of a 25-year-old here, you might have around 17,000 afferents, touch receptors, whereas by the time you're 75, you will have lost a good 4,000 of them. So you'll have only about 13,000. And this is why, as we get older, the tactile system doesn't function as well as it used to. And to put that into perspective, the 45,000 to maybe a million afferents you have in the somatosensory system is compared to maybe 100 million in a retina, or maybe 12,000 in the cochlea. So for touch, we're definitely talking about a big input here to the brain. So what I really focus on is touch. However, you do have to also take into account pain, so that's nociception, which we've already heard a bit about today, and also temperature as well, which is important. So I want to tell you about what I do, and this is a technique which is very specialist called microneurography, and we are the only team in France that do this, and it's very few in the world who do this as well. And what we do is we record from and stimulate a single tactile afferent, and that's in humans. So it's in vivo electrophysiology in humans. So here's just a schematic to show what happens in microneurography. We get a very, very small electrode, and we can just gently push it through the skin of a healthy person sat down. It doesn't hurt, and it shouldn't hurt. And what we can do is eventually we can reach a peripheral nerve, and we can then find a single touch afferent, a single mechanoreceptive afferent, and we can record from it. So you can see at the bottom, you get these really nice spikes, and there's a close-up of a nerve spike there. It's just like electrophysiology in animals, but we're doing this in humans. But what we can also do something slightly further than this is that, as well as recording from a single afferent, we can also stimulate it electrically. And this is how we're getting into the realm of artificial touch. So for example, I've got a tactile afferent which I'm recording from the end of the finger, and you can see a nice response when I press on it. What I can do then is actually back down this small electrode, we can apply a very, very small current, microamps of current. And so we can actually stimulate the very same afferent here and investigate the perception of how a single afferent feels in humans. So the first question is, does an activity in a single unit out of all these potentially millions that you have reach consciousness? And what we do is we'll stimulate it, for example, a couple of microamps for a second, and then we will ask people what they feel, how big it is, what the border's like, what the shape's like, if there's any movement. And so we can begin to quantify the perception of a single afferent in humans. And I've got some preliminary results in this. Hopefully it's going to be published soon, that in the glabrous skin of the hand, where you've got 4 different types of afferents, we recorded from 250 and stimulated 250 single afferents. And we find that for the different types of afferent, you get different sensations. So for example, with one, you get a small point of vibration, which actually relates really well to its receptive field properties. And then for a different one, we get the sensation of a small inwards pulling and a point of pressure, which actually matches again its physiology, because when you apply a pressure to this, it's called an SA1 Merkel afferent, then it will continue to fire. So that continuation of its firing matches very well with the sensation that's generated from when you stimulate it artificially. What we've also been doing is we've been trying to match the stimulating the single afferent in the periphery with the neuronal response you have in the brain. So we've been doing this with EEG and MEG, but we've also done it at 7 Tesla fMRI. So we've put some subjects into this very, very strong magnet, and then we put the electrode in their nerve, just like the normal microstimulation. And what we did is we stimulated during a protocol, and then we looked at the responses in the brain. So we wanted to link specific events in the periphery from what was happening in the skin, or we were stimulating the nerve, to with the simultaneous activation of the brain here. And so we had, as a comparison, we vibrated over the fingertips, so we could map the S1. And at 7T, this gives really, really nice detailed maps. So you can see here in different colors, the different responses from the fingers in the primary somatosensory cortex. And we can then compare that region to the stimulation of one single unit. And for us, it was quite amazing that actually you could see a response, and it was only seeing a very clear response here at 7T because of the good signal, but you could see a very, very small response that related to the sensation the person was feeling when you stimulated that single unit. And in fact, when you lost that sensation, for example, when we stopped the stimulation, that response in the primary somatosensory cortex goes. So this looks like it's the conscious perception of touch is happening here. And we can also compare this small sensation that's generated by the stimulation of a single afferent with something which is the sensation which is stimulated from by a point vibration at the site of the unit. And you can see on the bottom here that there is a lot more response here in the bold signal. So you can almost quantify some differences there between activating one single afferent compared to activating many different afferents. So how can we take the technique of microneurography and microstimulation and actually use it to generate artificial touch? So this is a project I've been involved with for a couple of years now, and it's something that's continuing. And what we want to do is we want to generate artificial touch in amputees. So there have been a few patients now who have been implanted with this. It's a special device, and it's made in Sweden. It's conducted in Sweden, these experiments. And you can see this person here has no arm, but what we can do is actually stimulate his nerve, and he can actually feel sensations from this, and he can discriminate between different stimuli. So already we're getting some kind of feedback from a prosthetic, which in itself has given him a lot more freedom. And it means that he can use his arm, and he embodies his arm a lot more than he used to. So this is how we close the sensory motor loop. And for the moment, we can, well, we're starting to be able to recover sensations like pressure and vibration. However, what I want to do is take this a lot further. And this is what I'm trying to say in the more distant future, but I hope it's feasible. That we want to recover not just the basic mechanisms in touch, but things like temperature and affective touch and things like this. And this is how we will build better prostheses. And so this man here, he actually works in the north of Sweden, and so he can still go about his job now. However, we want to provide him with the full embodiment and the full feedback that a normal person would have from their arm. So there are other things to take into account here. So another part of my work looks at other different afferents in humans. And we have something called a C-tactile afferent. And this is generally thought of as providing affective touch. And so what it is it responds preferably to the speed and both the temperature of a touch. But rather than, so what it does, it actually responds best to a slow, gentle caress, for example here. So if you go too fast or too slow, the responses are non-optimal, and it's the same for temperature. So if you stroke over the receptive field with a temperature which is too cold or too hot, again, its response is non-optimal. So what we think is actually these C tactile afferents are encoding the positive and effective, pleasant aspects of touch. And this is why we think they're actually involved in more social touch and effective touch here. And so to give feedback from a prosthetic into this type of touch would mean that the person could actually feel more of an emotional attachment to both their prosthetic and to other people. And another idea that I would like to provide prosthetics in the future is more complex sensations. So complex sensations would be, for example, when you touch a texture. At the moment, we really don't know how this is encoded, especially when you do it actively, because a lot of work has been done in the passive domain. However, so I want to look into this, as well as touch and temperature interactions, because what we know is that mechanoreceptors respond to touch, of course, but they also respond to touch at different temperatures in different ways. And so we can also move on to what I like to call pseudo sensations. So for example, this is touch and temperature working together. You get the sense of wetness. However, we don't have any receptor in the skin for wetness. We do not have a hygro receptor. So how is wetness coded? And this is something that we're exploring at the moment, because we know that through touch and temperature signals, people are extremely good at understanding and perceiving wetness. And I have preliminary results which show in the hand here, out of the 17,000 mechanoreceptors you have in your hand, and the 4 different types of mechanoreceptor you have, there is only one mechanoreceptive type which actually encodes, for example, drops of water. And we think this is more signaled by vibration. So to be able to give things like this back to somebody with a prosthetic really is the future, but I'm hoping it's a not too distant future. So I just want to end here by saying that touch is very important. You get significant input from all your afferents, and you can feel a single mechanoreceptor out of those millions of fibers that you have, which in itself is quite amazing. And it combines with all your other somatosensory signals to convey all that richness you have when your skin interacts with the external world. And I just want to say that both the discriminative and the effective touch aspects are very important, especially as this impacts on our health and well-being. So for example, touch, especially the effective touch, can strengthen our social interactions. And when touch is degraded, it can be debilitating. So we need to find ways to enhance touch and to treat and diagnose somatosensory disorders, and hopefully to provide that realistic touch back into a prosthetic as well. So I'd just like to say thank you to Neuro Marseille for this opportunity. And I really like this initiative, and I'm looking forward to the next ones as well. So thank you. Any questions?
Thanks a lot, Rochelle. You have one question from Pascal. If we link Lucie's work and yours, do you observe sex differences in terms of response when you stimulate nerves and record brain response using MRI? And do you observe differences in women dependent to estrous cycle?
Sorry, it's in the. Sorry, is there the text for the question there?
Yeah, I send you the text.
It's easier to see the actual text.
Do you see it or
sorry,
not?
This, it's the, I think the soutenance is finished. So, sorry, I can't actually see the question here. Is it in the question?
No, this one is not in the question, but if you go in the question, you have other one, like
okay.
you have from Benoit. Is there already a functional redundancy in afference coding?
Yeah. So is it okay if I answer maybe these questions first, and then maybe Pascal can put it in there, because it's a bit difficult to see with the screens and things. So I can start with the first question. Is that real? Is his 70 MRI system? Yes, we do use the same one. It was done in Nottingham, but it's the same one as we have here in Marseille. Yes.
The second one is for Lucie, and the third one is for you, from Benoit.
Okay, so Benoit. So is there already a functional redundancy in afference coding? Is it possible to observe two different subtypes of afferent coding for the same sensation? I don't think there can be redundancy in the encoding. However, it's true that you get for example, just at a normal touch, you will get various different afferents encoding it, but they do it in slightly different ways. And I think it's the way they blend together actually creates a sensation. So we're looking at individual touch receptors and their own responses, but I think that almost in touch, every single spike is important. And the timing between the spikes we're finding is very, very important as well. For example, if you really need to find information, for example, with grip, the timing is essential. So I don't think there's redundancy as such. I think that there is the very slight differences sometimes between the responses you'll get, and the brain is using all this information to actually perceive your sensation. I hope that answers that question. So will it be possible to make patients with forearm amputations feel the sensation of touching as if they still had their hand? Yes, that's what I hope to do. No, the sensation is not localized to the stump. It actually does localize to the phantom hand. It may not be exactly in the same position as before. However, we hope with a good prosthetic hand, which the person can embody very well, that the person will be able to localize the sensation to where it actually would be on the prosthetic hand. At the moment, we've only done a few patients, and this work is very new in the world, but so to be honest, it could be different between different people. But the person who we've been testing, he was amputated 10 years ago, and he feels the sensations when we stimulate in his actual hand, not on the stump. So this is a very, very positive thing for developing this type of technology. So next one by Anna. We don't have to learn to touch, but what do we know about tactile sensitivity in babies and their capability to explore the world with touch? Actually, it's very interesting that, as far as we know, touch is one of the, or if not the first system to develop in utero. So of course you need the skin to shape the baby in utero. So we have our mechanoreceptors, we have our sense of touch before we're born. We do know that babies feel touch as normal and things like that. However, I can't do microneurography in babies. We don't do microneurography in non-adults. So there's no way we could kind of go down that avenue. However, we do know that babies have a developed sense of affective touch. I have a paper which shows that the affective touch system seems to work very well in younger people, and there are collaborators who I work with who have looked using veneers in the brain to image gentle touch in babies, and the responses are similar to in the adults. So we think that the system is well developed in children and in babies, although for sure this system is going to develop when they grow. So there is room for, you know, if you don't get touched enough, for example, there is room for maladaptation of the touch system. which is something that is very important to consider. You need to cuddle your babies, that's for sure. So, next question. How can an amputee move the fingers of their prosthetic arm? And can they feel their finger move, albeit their artificial finger? Hopefully, working with other people in the world, this will be possible. So this is not my domain. My domain would be the signals coming in, the afferent signals. However, it's true that we still need to develop prostheses which are good enough to allow movements and have sensors in the fingers, for example. There are various projects going on at the moment into this and trying to capture what our mechanoreceptors capture realistically. And although this isn't my work is trying to understand the actual signals in humans. This is being developed in parallel. And so for starting, you can have prosthetics which grip and you can pick up things. And these are becoming more and more developed all the time. And so I think the additional question is, how can you get the movement feedback? So you can get the movement feedback from your mechanoreceptors because you're touching it, but it still lacks the muscle afferent feedback. And again, that is something that I would also like to look into in the future. Can you also give the person the proprioceptive feedback, which is something that we do in our group as well. We work on proprioception. And I agree, this is a very important point, that it's not just the tactile signals that you need your sense of embodiment, you need the sense of your limb in space and where it's moving and how it's moving, how fast it's moving. And so that's definitely another challenge there. Was there Pascal's question? Are there any other questions?
I think this is all. Yeah.
So can you hear me, Rochelle? This is Pascal.
Yeah.
Thank you very much for this great presentation. So my question was, if we try to link Lucie's work and your work, my question is, have you observed any differences between male and female? And for female, can you observe any differences when you do a stimulation and when you record directly in brain according to the astrocycle?
It's a very good point that I think all research in humans and animals needs to take into account the sex, gender aspects and the hormonal aspects. And for sure, hormones can affect your sense of touch. So it's not something that we particularly look at in our lab because we don't take, for example, females who are pregnant or who will have hormonal imbalances and things like that, because the hormones can change your skin. And so, for example, after the menopause, the skin can change. It becomes drier when you lack hormones, and this can affect your sense of touch. There are studies which have looked into touch over the menstrual cycle, and it can change. And as I said before, it can change over the lifetime as well. So it's actually important, and there are people looking at this at the moment. For example, during the menstrual cycle, are potentially females more receptive to touch, especially this affective touch, when they are kind of getting ready to have babies and things like this. So I think it definitely links in well with Lucie's work to do more with pain, but the whole somatosensory system for sure is affected by hormones.
Okay, thank you very much.
Thank you. Is there another question? Is there a difference between glabrous and hairy skin when studying water perception? And so would body hair increase vibrations and thus the outcome of the sensation? Perceptually, you do not feel a particular difference between the perception of wetness here on your hand compared to on your body. But we do know that actually the hairy skin on the body is slightly more perceptive to wetness. So for example, when you go out in the rain, you feel normally initially that the rain, for example, on your face, I think your face is a very, very sensitive part in the body, and you may put your hand out to try and feel the rain with your hand, but it's more on your face and on your lips that you're actually feeling these very, very small droplets of rain. And so perceptually, you don't always know this difference, but we do believe there is a difference between the mechanoreceptors that are encoding wetness on the glabrous skin of the hand compared to the rest of the body. And so I definitely agree that body hair can increase these vibrations, and the hairs on your skin definitely respond to, for example, drops of water, rain. And that is something that we do not have on the hands. And so although the skin on the hairy skin of your body is very sensitive, it's not very good at discrimination of touch. However, when it comes to the sensitivity, for example, in sensing drops of water or rain, it's actually very good. And this is in part just to do with your hairs as well. And another question, what are the causes of the reduction in skin receptor number with age? And can it be prevented? For example, would it be less marked in a masseur who constantly uses touch? Or maybe more marked because they're using their hands all the time? We do know that as you age, your body degenerates almost. And so this is just something that is the natural aging process. And the same will happen with your sense of smell and taste and hearing and vision. So it's something that is natural. When you use your system over time, it decreases. You can't keep this up forever. However, we don't know exactly what types of mechanoreceptor are decreased. We're not sure if it's just the myelinated ones, which send information very quickly to the brain, or the C fibers, for example, in affective touch or more implicated in pain and temperature. And there actually is possible that there are certain people who do not lose so many receptors, but we don't know why for the moment. It's just a finding in the past few years that actually some people, their sense of touch is very well preserved, but we need to do a lot more research into this to know why. And is there any way it can be prevented? We can't prevent the natural aging process, but I would definitely say for your skin, you can look after it. You can wear sun cream, for example. You can keep it hydrated, try and prevent dryness of skin, because if you have dry skin, this won't help your sense of touch. So you can use creams and things like this. I'm sure I could be selling creams here, but using a cream to hydrate your skin will give you a better sense of touch. And this is something you will probably find as you get older as well. Are there any more questions? I think
yeah, I think this is all. So thank you, Rochelle, for your presentation. It was very interesting.
thank you.
So I'm just gonna close this with the next diapo, and I'm gonna let Pascal just talk about the next Neurobinar.
So thank you very much. Can you see me? Not sure. It doesn't matter.
Yes. Yeah.
Okay. Thank you very much to all the speakers for this great presentation and for all the participants for interactions. So our next Neurobinar will take place in 1 month from now, the 19th of November at 4 p.m. again. And we will have 2 PhD students, Johanna Extremé and Yannick Becker, presenting their work. And then Clément Menuet from InMed will give the 20 minutes presentation. Of course, we will need more people to animate this session. If you want to present your work and share with us your science, you can contact Marseille and to be the next one for the December session. Thank you very much all for be here tonight. I hope you enjoy this session, and thank you again for all the speakers. See you